Okay, talking about DNA then, the first objective on page 19 asks you to describe the general structure and molecular nature of DNA. And this model that I have here is a model of DNA, and this is from your textbook. And we can see that DNA is in its coiled, tightly wound up form. We call that a chromosome. So we have 23 pairs of chromosomes in the nucleus of our cells, and that's simply DNA. So this DNA is tightly wound around these proteins called histones that keeps it in a coiled, almost like a spring-like structure. And if you look at this model, if I could coil this tighter, it would look like a spring. And then I can uncoil it and straighten it out and see that it's actually a ladder with the sides being sugars and phosphates and the rungs being the different nitrogen bases. So here's an example of an unwound DNA molecule. On the sides, again, we have phosphate and sugar. So in this model, these leather straps represent the phosphate and sugar, but that doesn't give us a whole lot of, familiarity with what those structures look like. So this diagram is a little nicer. The sugar is called deoxyribose. That's the name of the sugar. And then it's bonded to a phosphate, and those are covalently bonded. So the sides of the DNA ladder are held together by covalent bonds. Those can be broken, right, by digestion. Anytime we eat cells, anytime you eat plant or animal, you're eating DNA from that organism. So we're breaking those bonds and making new ones. The same side, or the other side, has the same structure, sugar, phosphate coming down, but notice that it's in the reverse order. It's almost like upside down. We call that anti-parallel. But then in the middle is the third part of a nucleotide, which is the repeating unit of a DNA molecule. The third piece is this nitrogen base. And there's 4 nitrogen bases in DNA: adenine, thymine, cytosine, and guanine. So depending on what information is coded by this DNA, we're going to see a different pattern of nitrogen bases. But they're always going to be these 4 letters. It's just the arrangement might be a little different. We might have AAC, CCG, whatever. And then the other side is always complementary to these nitrogen bases. So we can say A always bonds with T, because based on their molecular structure, adenine and thymine form hydrogen bonds with one another, and C always bonds with G, forming hydrogen bonds. So hydrogen bonds are the weakest of bonds. So that means this DNA can be unzipped and the inside nitrogen base is exposed to make copies of DNA. Anytime we need another cell, by mitosis, we need copies of these chromosomes so we can send those chromosomes to the new cells. So we're going to talk about DNA replication, which is just making a copy of DNA by unzipping or breaking these hydrogen bonds. So some terms now. Pentose refers to the sugar. If you look at the sugar, there's 5 carbons. Each little corner on this house-looking structure, that's a pentose, meaning 5 carbons make up that sugar. And the sugar is called deoxyribose when we're talking about DNA. And again, the 4 nitrogen bases are shown there. So if I look at the nitrogen bases in a little bit more detail, we can see that A and G, adenine and guanine, have a little bit more of a complex structure. We call them purines. And the thymine and cytosine, more of a simple structure, we call those the pyrimidines. And these terms, purine, pyrimidine, deoxyribose, these are going to be review terms for the worksheets that we're going to do at the end of lab. So just kind of highlight these in your lab packet so you can find them quickly. I recommend bringing a highlighter to lab so you can interact with your lab packet. And then if we compare that structure to RNA, RNA is just simply like 1/2 of a DNA ladder. There is no other side to it. So we still have the phosphate and the sugar covalently bonded together, and then we have just these nitrogen bases kind of just hanging out exposed. They're not hydrogen bonded to another nitrogen base. They're just out there. So this is RNA, a single-stranded. And the reason why it's called RNA is because the sugar is ribonucleic acid. So the sugar is ribose. It's just a little bit different than the one in DNA. In DNA, it's called deoxyribose. So when you write out the word DNA, what that stands for is deoxyribonucleic acid. The deoxyribo tells us the sugar that's present in that nucleus, or in that molecule. So a gene then is just a portion of a chromosome that codes for a specific protein. So if we go back to my original structure here, this area right here might have a specific pattern of nitrogen bases that codes for a specific protein. So we call that a gene, and that's a functional part of the chromosome. That's the part we need. It has the recipe for making a protein. And what are some proteins we learned in lecture today? Actin and myosin. Others? What? Troponin. Yeah. Other proteins you can think of? Collagen, structural protein. Yeah. So in order to make those proteins, we need the recipe, and the recipe is found in DNA. So that portion of a chromosome that has the DNA recipe for making that protein we call a gene. So we've identified genes for breast cancer, right? That we know the specific code, the specific spot on the chromosome where that breast cancer gene is located that makes someone susceptible to breast cancer. We've located the gene for cystic fibrosis. We've located the gene for a lot of inherited disorders. So understanding how DNA relates to protein function is the goal of our lab. So looking at the first process is during interphase is when DNA makes a copy of itself right before it enters mitosis and multiplies that cell number by 1, right? Because mitosis is making a copy of a cell to get 2 cells. So in order to unzip the DNA molecule, we need to separate the nitrogen bases. Wouldn't you agree? We need to separate these to separate the DNA ladder. So what type of bonds did we say hold the 2 sides together in the middle? Hydrogen bonds. Those are weak bonds. So we have an enzyme called helicase that's going to come through and unzip these hydrogen bonds and expose those nitrogen bases. So then DNA polymerase is an enzyme that's going to bring little nucleotides in here. If we look at this diagram in your textbook, the purple represents the sugar and phosphate, and the different little colored sticks represent the nitrogen bases. So depending on what's exposed here, what nitrogen base is exposed, the pair that matches that nitrogen base is going to be put in its place. So if this one here is adenine, then what nitrogen base would go here? Thymine. So we're just going to do base pairing, making sure the A's goes with the T's and the C's go with the G's. And essentially, you're making a new strand on both sides. So the benefit of that is when we're done, we've got 2 strands, but each of those strands has a parent strand and a new daughter strand, right? Would you agree? If I look at this, if I unzip this, I'm making a new strand here and a new strand here. So I'm going to have 2 DNA molecules when I'm done, but each molecule has the parent strand in it. And what that does is that preserves that code and allows us to just make a simple copy rather than recreating an entire molecule, which might be more error prone if we did it that way. So we call this process of DNA replication semi-conservative because there's a parent strand and then there's the new strand in each molecule. So if I look at it like this, here's my DNA molecule. I've got, when I'm done and it's unzipped and DNA polymerase adds new nucleotides, I've got 2 new strands, but I still have my old strand as part of those molecules. So when we need to make a protein, we talk about the process of transcription and translation. Transcription is when we're taking that DNA strand, we're unzipping it again, just like we saw in DNA replication, but instead of making more DNA, we're going to make RNA. And what did we say is different about RNA in terms of nitrogen bases? The sugar is ribose, but in terms of dropping nitrogen bases here, what nitrogen base is different? Yeah, the adenine is going to bond to the uracil. So anywhere we see an A, we're going to put a U there, a uracil, instead of a thymine. So uracil drops in, and we make a strand. And what did we say about RNA? Is it double-stranded or single-stranded? single-stranded. So here we can see a messenger RNA strand being created in this process of transcription. We're using a DNA strand to make an RNA strand by this base pairing, this A with T, or I'm sorry, A with U, C with G. Then what that mRNA strand is going to do is it's going to leave the nucleus via the nuclear pore and come out and take part in another process called translation out at the ribosome. So here's my DNA. It's unwound. mRNA is made by RNA polymerase, dropping more nucleotides in and bringing that code out of the nucleus to the ribosome to code for amino acids and build a protein. So this stranded pearls, row of pearls here is my protein. Each individual pearl is an amino acid. And the worksheets that we're going to do in just a few minutes are going to make sense of this for you. So if this is kind of like, ooh, kind of fuzzy, that's okay. We're going to build this little by little. So let's say this is my DNA, one side of my DNA ladder inside the nucleus. In the process of transcriptions, I'm going to make a messenger RNA strand. So T, instead of T, what would be my messenger RNA be? A. We're just going to do base pairing, A with T, C with G. So we're going to have A, U, G, and then just keep going, base pairing all of these letters until I formed my messenger RNA strand. And what enzyme forms this mRNA strand? Do you remember what we called the enzymes that are building these new strands just by base pairing? Polymerase, yep. So DNA polymerase, if we wanted another copy of this, that would be called replication, and DNA polymerase would work on that. But when we're building a messenger RNA strand, it's RNA polymerase. Then, when we get out into the cytoplasm, like I showed you here, translation, we have another type of RNA that is part of the process, and it's called transfer RNA, and it contains complementary nitrogen bases that match the messenger RNA strand. So instead of A, transfer RNA has U, A, C to go with A, U, G. And this sequence of nitrogen bases corresponds to a particular amino acid. So in this case, the amino acid is methionine. This set of nitrogen bases on transfer RNA codes for proline. And how do we know this? Well, there's a simple table in your textbook where you can find this information. If you know what the messenger RNA set of 3 bases is, you can figure out what the amino acid is. So these are the different amino acids, and these are the different nitrogen bases that code for that particular amino acid. But for this table, most tables that you use for this want you to use the mRNA nitrogen bases. So any set of 3 bases going in order from left to right is called a codon. So this is a codon. So there's codon 1, codon 2, codon 3, codon 4. It's just a set of 3 nitrogen bases on a messenger RNA strand. And that is complementary to the anticodon of transfer RNA that carries that amino acid. So AUG is on the messenger RNA strand. If I go to this table, AUG corresponds to methionine, which is a start codon, which means here's the beginning of my protein, start there. Oops. So that's what this is we're starting the protein here, and then all the amino acids that follow it are unique to that protein. So this particular protein might be insulin, or if we change the order of amino acids, it might be hemoglobin, right? So any different protein is based on what the order of amino acids are for that protein. So could you see that if I change some letters here, that might change letters here, which might change here and might change here? So any mutation that changes the DNA sequence of nitrogen bases can affect my protein and change its function. Because if I change the order of amino acids, I change its shape, like we talked about in lecture, which would change its function. So some inherited diseases are the result of mutations that occur in a protein during this process of DNA replication and protein synthesis. Okay, so there's a nice animation here that I'm going to kind of walk you through that shows how all these pieces fit together.
Here is a cell. The base...
I'm going to turn that off, though. Okay, so we've got a cell. Inside the cell are the different organelles. There's the nucleus. What would we expect to find inside the nucleus? Say again? I can't hear you. Nucleolus is another organelle that we would find inside the nucleus. But in terms of DNA, RNA, which of them is stuck in the nucleus? DNA. It's the only nucleic acid that can't leave the nucleus. So DNA is there. And DNA, when it's tightly bound like this, it's called a chromosome. When we unwind it, that's when we can use these processes of DNA replication and transcription and all that. So there's a chromosome, which is simply DNA. These are 2 strands of DNA. So now if we unwind that DNA, we can see there's the proteins, the histones. Here we can see the DNA ladder starting to unwind. What are the colorful rungs? What part? What part of DNA are the rungs? What? Well, the nucleotides are a 3-part structure. It includes the sugar, the phosphate that's found on the side of the DNA ladder, and 1 nitrogen base. So all of these colorful rungs in the middle here are all of the 4 different nitrogen bases. Okay, and like this one, maybe let's say this is adenine, the red one. What would the green one be on the other side? Thymine. Right, right. So this is the DNA ladder. It's being unwound, and it's highlighting. Notice that the animation is highlighting this portion of the DNA ladder. So we call that a gene. Tell me, do you remember what I said a gene was? What makes this highlighted portion special? Yeah, it codes for a specific protein that has a specific function in the body. Because some parts of our DNA don't code for anything special. There's no information on that part of the chromosome. So that's nothing special. The genes are where the information is that we need. Okay, here comes my big globular enzyme. That's a protein. So enzymes, we said, are proteins. And it's unzipping. What is it? What are the bonds that hold that ladder together that are being broken here? Hydrogen bonds. Hydrogen bonds. Covalent bonds hold the sides together. That's what's between the phosphate and the sugar on the sides. So then this enzyme, what do we call that globular enzyme again that's dropping these nucleotides? Any enzyme, RNA or DNA, that's building a new strand, we call that polymerase. Poly means many. It's adding many nucleotides to this existing strand to make a new strand. So in this case, we're making RNA because we're making a single-stranded nucleic acid, right? And you can see we're dropping U's instead of T's. So here's an A. If it was DNA, I would drop a T, but I'm making messenger RNA, so I'm going to put a U there instead. And then what type of bond occurs here between nucleotides, between the sugar and the phosphate? What? Covalent, yep, covalent bond. So the sides of the DNA ladder are stronger than the middle of the DNA ladder. Because of this process of replication and transcription, we need to be able to unzip these molecules more easily with less energy required. Covalent bonds are a little stronger. Okay, so we can see those nucleotides are dropping in. Again, a nucleotide is a sugar, a phosphate, and a nitrogen base. So in this animation, here's the nitrogen base, adenine, and the blue is, it doesn't show the detail, but it's actually a sugar and a phosphate bonded together. So we can see it's being unzipped, and this messenger RNA was formed, and now it's going to leave the nucleus and go out to the cytoplasm. And how does it get out to the cytoplasm? There's little holes in the nuclear membrane called nuclear pores. And what we saw happen here is there are parts of this messenger RNA that are not important for making that protein. So those pieces are spliced out. So that disappeared, and those 2 pieces were put together. So that nonsense part of the DNA molecule that didn't code for a protein, that was not a gene, that gets spliced out. And this is very molecular, way beyond the scope of our class, so we just kind of take it at face value. So there goes our messenger RNA. There's a ribosome out in the cytoplasm, and that ribosome contains RNA as well. Here comes transfer RNA, which is found floating around in the cytoplasm associated with these amino acids. So again, what are these three things? Yeah, nitrogen bases. And these three nitrogen bases code for this amino acid. So again, we go to that chart in our textbook, look for what three nitrogen bases are these and what amino acid does that represent. So that transfer RNA, in this case is UCA, so we can look that up in our book and find out what amino acid that is, and that's added to the messenger RNA. But notice that once it brings that amino acid to the ribosome, notice how the transfer RNA leaves. The messenger RNA strand is no longer necessary after that amino acid is brought to the ribosome, right? That piece of the messenger RNA strand just continues down until we get to the end. And when does this process stop? What are these little pearls again? What? Monomers. And what are the monomers specifically? What are they? Amino acids. So these are individual amino acids making up the protein. And the protein we call the polymer because it's many amino acids, right? So monomer, polymer, those are generic terms. But specifically, if we're making a protein, we say the monomer is amino acid and the polymer is a protein. And what bonds hold these amino acids together? They're a covalent bond, but what type? Peptide bonds, yep. And there is my protein. Notice how it's folding up and forming a very specific shape. So that protein might be troponin, insulin, glucagon, all the different proteins that exist, collagen, keratin, actin, myosin, whatever. So where do we get the recipe then for making proteins? Where do we look? What's the starting template for making a protein? Yes. That's it for today. We're done. No. Yes, that is correct. DNA is where all the action is. It's the recipe. So if you want to make chocolate chip cookies, you go to your recipe, right? If you change your recipe, would that change your cookies? Definitely. Add too much salt, not enough salt, it's going to change things. Same thing with DNA. If we change the sequence of DNA because of something that's inherited or something that is due to exposure of something in the environment, that's called a mutation. So a mutation is any change in the sequence of DNA, and that might cause a mutation. But what happens if that mutation happens on one of that non-essential parts of the DNA molecule, what it's not on a gene? Is that going to cause a problem? No, it gets spliced out anyway, right? So not a problem. So those mutations are silent. Some are harmful. If it's a mutation that causes hemoglobin to have a funny shape, well, now that person has sickle cell anemia, they can't transport oxygen very well in low oxygen situations, and they suffer disease from that. So that's a harmful mutation. Sometimes a mutation can be a benefit, like some people that have, what was it? There was a person early in the AIDS epidemic that had some mutation that made them resistant to the AIDS virus, but they could transmit it. So they had the HIV virus in their plasma, but it wasn't attaching to the T cells and causing AIDS. So that was an example of a beneficial mutation that prevented that HIV. And if we could figure out what that mutation is and induce that with a vaccine, we could prevent AIDS, and that's where all the research is, focused. But we do have gene therapy for certain diseases that we can actually insert a functional gene in place of the dysfunctional gene, but that's a whole area of biotechnology that I'm not familiar with. So anyway, different types of mutations that you need to know. A point mutation is where you take just one nitrogen base and a different one is inserted in its place. So it's just at one point in the gene where there's a change or a mutation, so a change in that nitrogen base. So that's called a point mutation. An insertion is when you add an extra nitrogen base in there, And that pushes everything else down. So if we add CCG in the middle of this strand, so here's my original strand, here's my new strand, it's much longer, and the pattern of nitrogen bases that code for that specific amino acid is going to be much different. So insertions are much more serious mutations than a point mutation might be. Because if we go back to this chart, if I have a point mutation that changes a C to a U, that results in a change of a C to a U, that's still proline, right? So that we could have changes in single amino acids, or single nitrogen bases here, and it wouldn't change the amino acid. So not all point mutations are harmful or change the protein. An insertion is much more serious. A deletion, same thing. If I delete these 3 letters and my new strand looks like this, that's going to definitely change the pattern of amino acids when I get down to translation. So some mutations are inherited, which we call those germline mutations. So those are usually found in, or they're always found in every cell of the body. So if the sperm or egg that was used to form you contained a mutation, we call that a germline mutation, and all the cells of your body are going to have that mutation. Or if that mutation happened early in embryonic development, when you were just made up of a couple of million cells before you became a fetus and a more developed embryo, if you had a mutation happen in that early developmental process, again, we call that a germline mutation, and all of your body cells at birth would have that mutation. A somatic mutation is when you have a mutation that occurs after birth and affects only certain cells. So for example, person smokes their whole life. They develop a mutation in the lungs as a result of exposure to the carcinogens in cigarette smoke, and they develop cancer, a mutation in the cells of the lung leading to tumors and lung cancer. That's a somatic mutation. So somatic mutations are acquired, can be acquired by exposure to mutagens. Sometimes we have mutations that occur during fetal development. And young children, babies, can develop cancers and mutations that affect function. But still, those are somatic mutations. They're just located in specific areas. And we find that those areas of the body, those tissues that reproduce a lot, like epidermis, epidermal tissues, and glandular tissues are always being replaced throughout our lives. That's a lot of DNA replication and adding in new nucleotides. And DNA polymerase can be a little sloppy. And mutations happen during that replication process, resulting in a mutation and resulting in cancer. Some people have immune factors in place that if a mutation occurs, there's immune factors that prevent abnormal cell growth. Things like tumor necrosis factor, which are molecular, smaller molecules that are beyond the scope of this class, but parts of the immune system that prevent irregular cell growth as a result of mutations. And there's a term you need to know in today's lab called apoptosis. It's on page, let's see, it's in bold, bottom of page 22. It looks like it's apoptosis, but the second P is silent. It's called apoptosis, and that's where the immune system, if there's cells that have acquired too many mutations, it's programmed to be destroyed. And those cells die. And that prevents them from growing and causing cancer. So some people's families tend to have more powerful immune systems, and they can better control cancer growth, where other people are more prone to cancers. And maybe you'll see that in your families, that you have certain types of cancers that run in the family. That could be because of germline mutations that have been passed down within the family. And I know you could even get tested to see, like certain families that have breast cancer, they can test to look for that BRCA gene that increases the susceptibility for cancer. So anytime we have abnormal cell growth as a result of mutations, we call that neoplasia. Neoplasia means new growth, but it's abnormal growth. So a neoplasm is a cancerous growth. Or it's in, it's every reference I've seen in the healthcare setting of a neoplasm is cancerous. I usually see them in terms of brain tumors. We often call those neoplasms. If something causes a mutation, we call it a mutagen. But if that specific mutagen causes cancer, not just a mutation, but results in cancer, we call it a carcinogen. So anything that's a known carcinogen, obviously we want to avoid that. Like we know cigarette smoke contains carcinogens. So for the rest of lab, what we're going to do is we're going to work on these worksheets and practice your hand at DNA replication, transcription, translation, using charts to find out the right amino acids, looking at mutations, finding what type of mutation, and then that'll be it for today. So I'll grab those worksheets.